Now we move to microarray. In each type of cell, like a muscle cell or a skin cell, different genes are expressed, turned on, or silenced, turned off. If the cells that are turned on mutate, they could, depending on what role they play in the cell, trigger the cell to become abnormal and divide uncontrollably, causing cancer. One way they do this is to use a DNA microarray to determine the expression levels of genes. When a gene is expressed in a cell, it generates messenger RNA, mRNA. Overexpressed genes generate more mRNA than underexpressed genes. This can be detected on the microarray. Red spot shows that gene was strongly expressed in cancer cells. Green spot shows that gene was strongly repressed in cancer cells. Yellow spot shows gene was neither strongly expressed or strongly repressed in cancer cells. Black spot indicates that none of the cDNA has bonded to the DNA in the gene. The first step is to collect healthy and cancerous tissue samples. Once the tissue samples are obtained, mRNA is isolated from the samples. mRNA is color-coded with fluorescent tags and used to make DNA copy. The mRNA from the healthy cells is green, meanwhile in abnormal cells is red. How DNA microarrays work? DNA copy that is made, called cDNA, is then applied to the microarray. The cDNA binds to complementary base pairs in each of the spots on the array, hybridization. Each spot will appear red, green, or yellow when scanned with the laser. There are 3 applications in microarray, which are 1. Cancer, 2. Antibiotic treatment, 3. Oral pre-cancer. Cancer. Genomic DNA is fluorescently labeled. Array-based comparative genomic hybridization used to map genetic abnormalities in a wide range of tumors. Gene expression data can identify groups of cases with significantly different outcomes. Antibiotic treatment. Increase in number of resistant bacteria led to failure antibiotics. In oral cavity anaerobic bacteria, they are not easily culturable. DNA microarray analysis help to outlast the viability of the bacteria and a diagnosis can be made. Oral precancer. Leukoplakia or white lesions of the oral cavity may result from a myriad of reversible conditions. Identification of gene expression profiles allow to differentiate harmless white lesions from precancerous lesions. Early diagnosis and management of oral cancer is correlated with increased survival. Single strand of DNA of gene of interest can be immobilized in a grid or array, which then will be either glass slide, quartz wafer, and a nylon membrane. For example, in tumor biopsy, mRNA is extracted, labeled and hybridized with the array.
Created using Powtoon.